Okay, great. Good morning, everyone. Welcome to the panel discussion for Symposium 1, Section 4, Development of Quantitative Comparative Approaches to Support Complex Generic Drug Development. I really hope you enjoyed the wonderful presentation this morning. Before we begin with the panel discussion section, I would like to introduce 3 additional panelists that they will join our speaker for this panel discussion. The additional panelists are Dr. Darby Kozak, is the Deputy Division Director for the Division of Therapeutics Performance 1 in the FDA Office of Generic Drugs. Dr. Kozak leads the group of interdisciplinary scientists on the development of the new analytical method and the equivalence evaluation methodology for complex drug substance and the parental ophthalmic and inhalation formulations. Dr. Meiyu Shen is an expert mathematics statistician and a team leader in the Division of Biometrics 6 in Office of Biostatistics at CEDAR. Dr. Shen has published more than 50 papers in the Statistic, Medical, and Engineer Journal, and 3 book chapters. And Dr. Liang Zhao is the Director of the Division of Quantitative Method and Modeling, Office of Research and Standard, OGD and CEDAR. Dr. Zhao has established his leadership in industry research and drug development, quantitative method and modeling, and the model-based strategic decision-making in regulatory and industry setting for generic and new drugs. And Dr. Zhao will also give a closing remark this afternoon. A big welcome to all the panelist members. So I would like to remind our audience to type your questions in the chat box. We will try to answer as many questions as possible. So let's start. So the first question, what will be a guiding principle on applying quantitative statistical analysis for API sameness at the absence of clinical data from bioequivalence perspective? So I'm going to direct this question to Dr. Liang Zhao. Liang, you're muted.
Can you hear me now?
Yeah.
Sorry, I just switched my computer. There's some clumsiness in terms of switching things. It's a very key question for the session. My preliminary thinking is, there for any methodology development, there are 2 thoughts. It's more like type 1, type 2 error kind of a principle. First thing for the agency, we don't want to prove any product that have shown different efficacy safety profile from the ROD. That's the thing we want to guard and control. It's kind of analogous to a type 1 error. We really don't want to approve any product that can lead into a different benefit risk profile. The other thing is that given the lack of clinical data, for most of the cases, like which component is contributing to the efficacy and which component will lead to the safety concern. So we do want we we are developing some statistical approaches on a quantitative methods to evaluate the sameness. However, we don't want to make the method super sensitive that can pose some insurmountable challenge to the applicant, to the industry, that that make the cost very high and basically make no product that could pass. Then we need to also leverage the prior information, more like adopt a Bayesian type of thought, how we learned, or what we have learned from other complex APIs. What are the associated risks? We need to have a very good assessment on the risk if we are proposing a relatively relaxed approach to allow the reasonable path in the development. Those are the two thoughts from me. I believe other panel members will chime in for this important topic as well. Thanks.
Thank you. Dr. Tsong, do you want to add your comments as well? I think you're muted.
Yeah, I think basically when developing statistical method, we need to first develop the matrix to measure the difference or similarity between the test and the reference product. Then you need to derive estimate to that matrix, and then you need to find a margin. So typically there's a challenging, because when you develop the margin, you have only limited reference product to estimate. When so when after you develop the method and the margin, when it's generalized to a more generalized application, then you probably have to revisit whether the margin is. And also, many of the situation, as we see in the distribution profile comparison, the estimate usually is a biased estimate. For example, F2 estimate is a conservative as to the true one. So once you pass the using the deterministic approach, you definitely pass. But if you are failed, you probably wanted to do the bias correction and also bootstrapping to see that you can pass. So there's a lot of difficulty of developing methodology for this in the new area, new application, that we have to be, keep on working on that to especially when we want to generalize it.
Great, thank you. So Dr. Meiyu Song, do you want to add your insight?
Yes, I think there was if we don't have relevant clinical data for information, maybe we need a little bit more sample size, a little bit more data to understand the product quality, how to compare it. Because if we don't have clinical relevance data and we don't have more data on the product quality, it's really very hard to do statistical analysis. We really don't know what the uncertainty comes from the data, the product. So I just emphasize, if possible, get a little bit more data. I understand the CMC area, we only have like 6 batches or 6 data points, 10 data points, but really to validate the statistical inference, we need a little bit more information to understand the uncertainty of the product.
Great, thank you. Those comments are very helpful. So we'll move to the next question. So, so there are many new publications that provide newly available equipment and the method to test the API sameness. How does the agency evaluate which publication and analyze are relevant from the point of view of public health? So I will start with Darby.
Thanks, Dr. Ren. I think this is actually a great question, because as scientists, we're continually pushing the boundaries of what we're capable of doing. And a key component of that, I think from an analytical perspective, is developing both new methods to make new measurements, but also importantly, improving the sensitivity, accuracy, and precision of those measurements. And a key component of that is that increased accuracy and precision gives you greater confidence in that measurement. And so what that means is we're developing those methods to do so, we can potentially not have to do other methods or other sort of tests that may have less sort of accuracy or sensitivity. I think a key aspect there is when selecting that, I mean, we're always advancing, there's always been a new method out there, is now then thinking about your drug product and the API and the other sort of critical components of it, and best fitting that method to that unknown question. Okay, so if it's the molecular weight distribution or something like that, it's choosing and selecting an appropriate method for that. You may not need the most advanced system to look at a fairly narrow molecular weight distribution, a linear sort of polymer. But now let's say if it's got something of higher sort of complexity to it, there can be aspects there, still unknowns and gaps that not are being addressed by the current method that may be better addressed by other methods. So I think that to me is a key component of what is still the unknown that we are still need to develop and understand better, fitting that method to that, selecting that method appropriately. And like I said, is also there can be orthogonal methods. And that's, I think, one of the components of FDA is always asking for is, now, getting that orthogonal method, how does it complement that existing method? Like I said, if you're not necessarily using the most advanced method that can characterize all of it, you can use two separate methods that can get it some pictures from both sides. This might kind of get back to Francis. I really liked in your sort of talk that traditional image of the elephant. Do you mean you can use either a wide macro lens that can capture everything highly sensitive and see the whole thing, or you can get all those different components together and combine them together. And I think to me, that's kind of the aspect of selecting those methods there and using it appropriately, sort of using the research appropriately and the new methods that are out there. We're always going to want you to use the most accurate and sensitive method, but at certain points, you can equally justify, hey, look, I've captured this through multiple components. This gives me the confidence that this is the structure I've got or the component that I got. So I hand back, but I think that's, to me, is a really exciting sort of question.
Thank you, Darby. So Francis, do you want to add your insight from industry perspective?
No, absolutely. I think Darby's hit the thing on the nail. We've seen this all too often where, say, for example, I've done a lot of work, spent 2-3 years developing a method, used a, say, 500 megahertz NMR machine. And, I've seen resolutions, and you get minor structural, resolution where you can see even signals that are 0.1%. You've done this extensive work identifying various components of the product. And
so, Francis, we lost you.
hello, can you hear me?
Yeah, we can hear it clear now.
Okay, where did I get cut off? I'm not sure where you last heard me.
The last I heard, Francis, was on the 500 megahertz, where you're saying that you've been able to get down to 0.1%.
Yeah, so say I've done, I've gone down to 0.1%, and, I'm all happy because at this level, I've established equivalence, at least in my perspective. And tomorrow, I look at a new paper that has used a 1000 megahertz machine. And now all of a sudden you see minor components that you would not normally otherwise see. So now if I, first of all, without having the benefit of doubt, are these components really components or artifacts? Okay. So then what do I have to do next? This 1000 megahertz, if I even want to see whether these components are present in my product. I then have to go and find a machine like this, which probably exists only in a handful of places across the world. Then find experts who can probably do the work on this. Then develop a method. Sometimes if you have such exquisite articles and publications, it takes maybe 1-2 years if you're really inefficient to get this sort of a method established. And then you don't know because sometimes these signals, how small are they and how relevant are they? Because do they really matter at this small level? Because sometimes what we really look at is, sometimes as industry, we need to understand what is the role of these papers. Are they advancing science, or are they advancing generic drug development? And how do they fit in? You have to draw the borders between generic drug development, between advancing science, and basically public health policy. So this is really something that I would like to have your perspective also. How does health policy fit into this?
Yeah, Kia, you all right if I respond a little bit?
Yeah. Go ahead, Darby.
No, I think it's a great comment, Francis. I think that to me is a key aspect there. I think it's coming down also to the justification. You can always, as you get more sensitive, be able to find potential differences. So especially from a generic, I think you've got a couple different aspects that we potentially can look at. And one thing is, one of the beauties of the generics is you always have that reference product, multiple lots of the reference product, and that can actually help you set some of those justification limits. And I know it then becomes an additional sort of challenge, especially when there's heterogeneity, even batch to batch of the RLDs, which batch and which lots of RLD that you get, that you test, then kind of defines sort of that sort of design space that you get. And obviously, the more that you get, the design space becomes a bit more sort of clear to you. So that's one advantage that you potentially have. But I think the other sort of key aspect there is now looking back at those relative percentages and taking more of that, like you said, that sort of step back, maybe a holistic sort of view with the other methods you got, and looking at it saying, yes, if we microscope just down into this one area here, we might see a small sort of differences. Here's some of the other sort of components that gives us that additional sort of confidence that overall, on the broad picture, this active ingredient or this composition would be considered equivalent or the same. I don't think it's necessarily direct, but it kind of gives you, like I said, is the thing that we're playing with, both from the regulatory perspective, looking at those justifications, and also better understanding which methods we potentially may want to look at and use, and complementary. So, but thanks.
Now, just one last comment on this. So when you initially spoke, you spoke about this totality of evidence. When you spoke about this elephant thing, you made a few comments about using orthogonal methods. So I think that is a very interesting aspect, because when I read through the approval packages for glatiramer and enoxaparin, one of the key facets that came out was not that a product is fully characterized, but that it is adequately characterized. So it is up to us to, I think, draw the line when you think a product is adequately characterized, and then not push beyond and beyond, so that the patients don't get the medication at the end of it.
Thank you both. Meng, do you want to provide your comment as well?
Sure. Thanks for the discussion. I think it's very the key critical issues to address, and I would like to add some kind of how we kind of deal with the newly published, the results, the method, something. I think certainly agency welcome the innovation, especially useful for the to facilitate generic drug development. And so for the industry, if you identify any new method you think is useful and critical for your development, and you want to communicate with FDA, then I think you can use like a country correspondence or pre-NDA to communicate with us, and we can start from there. And from our side, we're closely monitoring all the new method tools, including from, let's say, publication and the communication with the industry. And once we identify anything critically can improve the drug development, then I think from us, we will carefully evaluate the method and also even conduct some kind of validation on the method. And we are also very open to share what current thinking, like from multiple routes, like a presentation, like a publication. Even if we can finally validate and find something critical, useful, then we may even do the revised guidance. I mean the product specific guidance and the new publishing new the guidance. I think that's what I want to add.
Thank you. Thank you all. Those are very helpful. So we move to the next question. So how to inform clinical relevance for different component of complex API? So first, I would like to pose this question to Darby.
Yeah, thanks. This is a good sort of challenge and a good sexual follow-on to the last sort of discussion, I think. And it's got its own sort of inherent sort of challenges too, because with active ingredient or pharmaceutical equivalent, especially the active ingredient, you're always looking at the structural similarity between the aspects of it. I think the real justification additional, as we're saying, is if you start to see these more complex APIs where you have heterogeneity, both within the structure or the sort of composition within the components of a complex API, that you need to start looking at, okay, so what's the relative weight of evidence of these sort of aspects? And there can be things from the orthogonal in terms of, okay, so what aspect is critical that we ensure the matching of, and those things that, you know, they can have a bit more sort of flexibility of that sort of approach. I think the glutamate acetate's a prime example there. We've looked at it. You get appropriate sort of matching and aspect. From a clinical relevance, I think there is a couple things that potentially can start to help include in terms of the justification. Examples I think of immediately off in terms of is some of the heparin sodiums where you can include like a bioassay or an activity assay to help support that API sameness, that these complexes these active ingredients are inherently complex. To be able to match all those polysaccharides is exceptionally sort of, that match can be challenging, but you've got a good sort of activity assay that you may be able to use as supportive evidence to say that though that heterogeneity is present, you can actually see is that in terms of these in vitro sort of studies that you do have a potential sort of aspect to support that active ingredient sameness for the PD there. The other things I also think of is in terms of the mechanism action of that active ingredient to help support any sort of variability differences within it. Examples I think that was presented was the conjugated estrogen story, which is a naturally sourced from horse pregnant mare urine and then purified from that. So you have a whole distribution, a lot of different components of the steroids, as well as non-steroidal sort of low molecular weight sort of aspects. And when we looked at this, we've done multiple batches within FDA to better understand those sort of levels. We looked at it and said, okay, well, the steroid component is probably the main sort of mechanism of action there. So that's where we need to have comparable sort of levels within it. We need to make sure that we control the non-steroidal components to make those types of measurements, but they necessarily don't need to be comparable within the same sort of structure. So you're looking at those sort of aspects of saying, okay, so what is a key component here that you may want to use as part of the justification when you've got sort of variability and within the sort of which limits that you'll start to set, especially from a statistical perspective as we move forward. There's a lot of other sort of examples, I think, where you start looking at things of their activity, local GI acting products and things like that, phosphate binders, other sort of potassium binders. We're actually using studies of the in vitro sort of binding to better understand the overall active ingredient and bioequivalence perspective. And you're using the structural sameness to support that, and all that taken together gives you that sort of that holistic sort of picture. And I agree, like the complex APIs is one area where we need to start to look at a bit more from both a statistical as well as a comparable, what would be considered to be comparable, because they raise inherent sort of challenges.
Yeah, Kay, if I can make a comment, I think this is probably the most complex question. Nobody can give an exact answer, kind of $1 million question. But I would like to welcome Dr. Francis. Since I just checked your company information, you guys been doing a lot in terms of playing around with complex API. So in your experience, how would you, I think probably you guys more think from a quality perspective currently, but if the products manufactured from another side or from a different process, how would you control the final product, convince yourself that they are bioequivalent against each other?
No, I think that's a very interesting question, and sometimes it depends on what are our critical quality attributes that we have defined within this space. So if I say, hey, I need to have this particular structure in this particular quantity, and then it does not, I personally feel it does not matter which area or which route you take to reach to the endpoint, as long as what you have defined in the end is all there. As long as those structural signatures are all there, you can have certain process related signatures, which are, if there are process related signatures, of course, you need to have them in the final product as well. So I think really it depends on what your final release parameters are and what are those critical attributes that you are looking for in the end. If those are there, I think, and if you're able to justify and soundly present the logic for those, I think you can have a very good chance of showing equivalence between different processes.
Do you guys apply some kind of a statistical analysis at the end, even though there will be multiple components in the API? They may differ in terms, there may be slight difference in terms of proportion or sometimes even physical chemical structure. So so how do you kind of do you apply any quantitative standard at the end for that?
Yeah, that I would say that would come probably towards the end. The more important thing is to first, as you go through the entire chain, you have your physico-chemical things. Okay, at that level, you may have similarity. But really, once you get into the high resolution methods, that's when you can really make a decision whether this is good or this is bad. And of course, if there is not too much difference, you can always apply these advanced statistical tools to see if in those, is there anything else that comes up. If nothing comes out of that, then you can actually make a comment whether they're equivalent or not. So we do apply these sort of statistical tools at our end also. But when you're developing complex APIs, really, how much time do you have to develop different routes? You sometimes have to stick to one route only.
Okay, thank you.
Yeah, thank you. That's a very good discussion. So there's a follow-up question, like for in vitro study for API, the sameness, the assessment. Normally, the sample size is very small, less than 10 lots. So that will introduce difficulty in applying meaningful statistical method or test. So there's question, does the FDA have any input on that? So I'm going to direct this question to Yi and Meiyu. Thank you.
I think most of the statistical comparison method is comparing the mean. As you know that for the inferential, there's a, when you compare the mean, you need to have a standard error of estimation. The larger the sample size you got, the smaller standard error you're gonna have. So with sample size, say 3, you're going to have a very wide standard error, so very wide confidence interval, so you won't be able to establish sameness. So that's, even though you have a 3 batch, so we typically ask for 3 batch, you need a 10 measurement per batch, so make it altogether 30 measurement. So that help you to show the sameness of the test and the reference product. Otherwise, statistical method can, if really small, you have 3 sample size, 3 or 6, you really cannot do much statistical analysis to qualify for the sameness. unless you make a very wide margin. But widening the margin, it may not be acceptable for the regulatory purpose. So it's a difficult issue here, and so that's that we have to consider how we're going to be able to deal with a small sample size statistically.
Yeah, this is Meiyu, so I can I echo Yi's concern. So when we have very small sample size, 3 or 4, 5, 6 or something, so the estimation, the mean you estimate from the data is not really stable, can vary a lot, and the variability can be even variable. So to make a statistical inference, we do need a little bit more information to understand the product. So every observation is representative from the distribution, the population, but then we do have a little information, and then our decision rely on so-called statistic is not really that robust scientific, because we only have very limited information to understand the whole population distribution. So statistics is a data science. So we cannot do magic. We cannot do statistical magic. We're not a statistical magician here. So we rely on the data, the information to make a rational decision. Yeah, that's I can share right now. Mm-hmm.
Yeah, thank you, Meiyu and Yi. So next we'll move to Yuqing. Do you want to take over?
Yes, thank you, Ki, and thank you for all the interactive discussion here. So I'm going to lead to some questions we received from the chat box. Our first question is, it's direct to Meng's presentation. So regarding the use of EMD calculations, would it be acceptable to use EMD as a superior metric for evaluation of PSD when compare products, even if the draft PSG for the product indicates that D50 and span should be used? So we, the audience who asked this question, are working on the product where the R&D PSD is not monomodal and has a wide span. So however, the PSG for this product specifies that the span should be used in the in vitro BE assessment. So we have performed the in vitro BE analysis and found that EMD calculations indicate equivalence, but the span doesn't. It is not clear if we have the option to use EMD when the PSG does not indicate that it is an acceptable approach. So I would like to invite Meng to provide your comments on this one.
Right, thank you. I can take a first tackle, and the other panel member feel free to make a comments. So I think there's a general kind of a thinking on the method we use for the PSD evaluation. If let's say the PSD profile follow normal mode distribution, then we think the traditional D50 and span are sufficiently sufficient. So then if you see and you confirm the PSD profile, it's complex, like a multi-peak or multi-mode, then you can consider another alternative method like EMD approach. We already had a recommendation on some products. You can consider using that. So I think your question is for your product you are developing, the current PSG recommend D50 and span to conduct the PSD equivalence analysis. However, when you conduct your study and analysis, you find the PSD profile is not monomode. I think this question consists of two parts. First part is it's about your analytical method, whether it's suitable, whether it's a fully kind of or sufficiently validated. If you can confirm that, on top of that, then you can think of oh, so okay, I'm pretty sure from my perspective the PSD is not monomode, then what kind of a method I can use. So for the particular question, my suggestion is that you need to confirm your analytical method is fully validated. And if you still think it is, then I will, for this specific question, I will encourage you contact with us by submitting kind of a control correspondence, or if you fully prepared, you can submit a pre-NDA meeting. We can touch base with you regarding this specific question. Yeah, that's my response.
Thank you, Meng. I see no others are going to provide additional comments. Then we are going to move to the next question. So the next question is specifically towards complex injectables. In order to measure the discrepancy between particle size distribution profiles, FDA recommends follow to use population bioequivalence, PBE, as one of the statistical methods. But this is usually followed when the distribution profile follows a monomodal size distribution. However, when distribution showed an additional peak at smaller particle sizes, and in addition contain a shoulder indicating some fraction of the particle size, having distinct higher molecular weight than the average number given. So for this case, under those kind of cases, how would be the approach followed to measure the two peaks distribution for confirming the similarity? So I would like to invite Meiyu and Yi to provide comments on this one first.
So the guidance method assume a distribution, a normality, the normal distribution. Well, for if there are two peaks, we can use bootstrap to generate a confidence interval. So that was, or you can use non-parametric method. That was for distribution, not to follow unimodal or normal, we can do bootstrap. Usually is the way to do it.
Yeah, I think in the guidance, the PBE was probably defined for the clinical trial, for the PK study. And this is using the crossover design. So you needed to have a normal assumption to be able to derive all this statistical and confidence interval. But in the in vitro equivalence study, we have those parallel arm study. It's not a crossover. So we need to, for example, in the particle size distribution, we need to make the test product and the reference product into a triplet, so we be able to derive the ratio between the test and reference difference and the reference variability. So we don't necessarily need a normal distribution because you can derive that using the bootstrapping method to do it. So it doesn't have to be normally distributed for the in vitro, comparison.
Thank you. And Meng, do you want to provide your comments on this one?
Yeah, thank you. Actually, my comments is pretty much similar to the comments I made for the first question. It's this question mentioned that they see kind of a small peak or the shoulder on top of the main peak. I still highly encourage you first carefully examine and validate your analytical method. I think that's the guarantee about your fidelity or suitability of the data you use for equivalence analysis. Because our previous experience, I met a case. It's the applicant claimed they have a double peak. However, during our analysis, we find out they have some deficiency in their analytical method, like a dilution rate, something like that. So I think if everything really get confirmed to say, oh, there's no problem in the analytical method, we really see kind of a complex profile, then I think alternative method rather than definitive span should be considered.
Thank you, Meng. Darby, do you have any comments on this one?
Yeah, and I think Meng's kind of hit the nail on the head. And I come from more of an analytical perspective. I think one aspect there is making sure that the analytical method is actually appropriately developed, as Meng said, is if it's underneath a non-ideal conditions, sometimes you will get those types of artifacts as secondary peek in. So sometimes there you'll want to use an orthogonal method that may be completely different sort of component to it to confirm that yes, it is a multimodal distribution or non-normal distribution. And we have actually seen that a few, and FDA has done some really good sort of studies on it. And then you need to kind of justify what component, if the particles are actually the double distributed. So I'll give a couple of examples. So one example that we did is a really good is the MDRS for inhalation products. You've got both suspended API particles and lactose particles. Now, if you're just to put that into a laser diffraction system, you're going to see sort of a very broad distribution, predominantly done by the lactose, because that's the majority of the particles in there. You put that through a system like MDRS, which both uses imaging and Raman, you can then start to identify what are the actual API particles in there, and then getting that particle size distribution that may be more appropriate to actually applying your statistics to. Another study we did for injectables, I think that's kind of focused on this study here, is potentially when you're using things like excipients like carboxymethylcellulose. This also has an inherent sort of a size when in solution up in the micron sort of area there. So you can have both suspended particles that are drug as well as this excipient. So if you were to put it through some systems, you might see multimodal. One peak is completely dependent upon the excipient, the other being the active ingredient. So applying statistics to both the excipient and the active ingredient might not necessarily make great sense. What you want to do is then focus in on a method that can identify potentially the active ingredient, making sure that we've got control of that component there. So from a purely analytical method, when you start to see things that aberrations are multimodal, even when you're just characterizing, you know there could be different sort of components that are contributing to your particle size. Using methods and kind of exploring that and justifying that my method is actually fit for purpose to study the property of my intention, and then start applying that statistics to it. So I think that would be my biggest sort of suggestion there. And that kind of piggybacks off what Meng said is definitely start there. And then as you start to get into it, make sure we're applying statistics appropriately for it.
Thank you, Darby. And there is follow-up statistical question about particle size distribution. This is direct to E. So in a PBE study for particle size distribution, do you use log of each value for statistics? What is the rationale to decide if you use your raw data or transform to the log number? E, do you want to answer this one?
I believe when we developed the ratio of chi-square method, we didn't use the log transformation. It's using the original mass measure in there.
Thank you, E. So our next question it's about the bootstrap. So how does the bootstrap approach with replacement a single set of tests and reference compare if repeated across different 12 observation or product data sets from the population of 1,000 simulated test and reference profiles? I'd like to invite Meiyu for this question.
So I want to make some first clarify this question. What do you mean on single data set of test and reference? Is this mean we cannot tell this test and reference? So when we do bootstrap, we cannot put test and reference together and bootstrap it, because then we will generate an unbalanced data set for test and reference. We need separate test and reference, separate. And then we bootstrap test product or bootstrap reference product, right? And then we have to match. So there was different way to match the test and reference product, the profile. So is my understanding right?
Thank you.
Yeah. So 12 units from test, 12 units from reference, then we cannot bootstrap 24 units together, because then we create an unbalanced situation. We needed 12 units for test, bootstrap that, and bootstrap 12 units for reference, and then we have a difficult to match. That's we talk about bootstrap method for the profile comparison.
Thank you, Meiyu, for your answer. And we are having another question. If FDA now expecting bootstrap F2 modeling, so Qi, I would like to direct this question to you.
Okay, thank you, Yuqing. So due to the high variability of the data, so comparison of the mean profile with F2 may not sufficient. So F2, the confidence interval, is it recommended to use a bootstrapping procedure? Thank you.
Thank you, Qi. And I would like to turn the panel back to Qi to follow up some dissolution profile similarity question. Thank you.
Thank you. So this is a question for Youssef. For your slides, you mentioned about the question is the F2 bootstrapping always more sensitive than model independent, the MSD approach? Also, in one of your slides, you mentioned about the early time points. Could you please define early time points for the immediate release drug product and the extended release product? Thank you.
Yeah, sure. Hello, everyone. So actually, to answer this question, as you know, there are a few studies that showed inconsistent conclusion about the sensitivity of each method, the F2 bootstrapping, the model independent or model dependent MSD approach. So in this work, actually, we tried to conduct this kind of analysis from the applicant perspective, like how they do the analysis. And we, as you observed from the findings, we presented that the F2 bootstrapping showed a more conservative conclusion. So actually, the results we presented were related to only using F2 bootstrap. Currently, we are also working on the bias-corrected accelerated bootstrap F2. So the results we obtained are very similar to the ones we presented. So the BCA bootstrap was able to detect the dissimilarity between the reference and test profile at theoretical F2 less than 50. However, with the BCA bootstrap, we observed that at theoretical F2 from 51 to 55, the bias-corrected one, the passing rate decreased significantly. This also indicates how the non-bias corrected F2 bootstrap is actually could bias the results. As I think Dr. Tsong and Shen will also provide more details on controlling type 1 error in response to this question. But again, here, this work, we just try to do exactly what the applicants do from their perspective. So regarding the time point, the initial time points, I believe actually the current practice is to include all early dissolution time point in the analysis. However, we noticed that if there is more time points with zero release, this could actually increase the similarity conclusion, especially if the applicants are applying the model-independent MSD approach due to the decreased difference between the compared test and reference profile. Sometimes also we observe that there is the test and reference don't have the same time points, especially the early time points. So we could apply the model-dependent MSD approach because, as you know, the model-dependent approach doesn't consider the number of time points for each test or reference. But here the problem is to find the model that could fit both the test and reference profile, especially if they have different shape. Also regarding the early time points, it's important to consider the variability, especially if we have a very low release at early time points. You know that the CV percentage could be very high, but the variability actually is very low. This is because the very or significantly small release at early time points. So actually this could lead to more restrictive thresholds for variability for early time points than at later time points. So this is actually answer to your question. I think now Dr. Tsong can provide more details about
Great.
bias and controlling type 1 error.
Great. Thank you.
Yeah, sure.
So there's a larger question for you. Are 12 units are sufficient for the comparison, the dissolution profile for highly valuable dissolution data?
So actually, it depends on the results from F2 bootstrap. If we have concern regarding the F2 bootstrap results, then we make, especially to the high variability, it's good to increase the number of units. Like for example, 24. So we have a solid conclusion from the F2 bootstrap or the appropriate method used for the assessment of the solution and similarity.
Great, great. Thank you. So we receive another question about the communication with the agency. If I have question regarding the quantitative, the modeling and the data that analytical that needs the FDA's comments, what is the best channel and the most efficient way to ask through? So I will direct this question to Liang.
Absolutely. I think we have a standing controlled correspondence channel for single question. If I have a simple single question, we can turn that around in 2 months. If it's simple enough, then we have a complex controls, but still single question, then we need a currently 4 months to respond to the question. If the question is comprehensive enough and engaging multiple disciplines, offices, and it may compromise, you may have several questions to discuss with the agency. We are more than happy to accommodate a communication between us in the form of a pre-ANDA meeting, a product development meeting. If there are multiple other initiatives that have been in the GDUFA 3 commitment. So stay tuned. I will leave some other details to my colleagues like Darby and others to comment.
So Darby, do you want to comment on that?
I think just to kind of reiterate what Liang is saying, I think one aspect, especially if you've got a lot of different types of questions that scope across, the pre-ANDA meeting definitely is a great opportunity there. It allows us to collect all the subject matter experts across a wide range. You're getting analytical sort of feedback as well as modeling feedback. So a very comprehensive sort of approach. But I think it also comes down to, as Liang was saying, the types of questions you have and how focused they are, and potentially what sort of things that they touch upon. Yeah, I don't have any additional. I think that was a really good sort of response from Liang.
So Franz, do you want to comment on that from industry perspective?
No, I absolutely. In fact, Liang was speaking everything as I was imagining it. He was saying the exact same things because it really lies in the complexity of the question itself more than anything else. So if you have multiple disciplines that are needed to take care of a question, then you're better off having, apart from the goal dates of the control correspondence, then you're better off having a pre-ANDA meeting. And sometimes I think the pre-ANDA meeting, what we, in our experience, we've used the pre-ANDA meeting to kind of highlight or rather look at the weakness of our approach. And then if you have control correspondences later on, based on the inputs received in the pre-ANDA meeting, maybe you can get more directed and appropriate responses later on.
Great. Any other comment from panelists? Okay. If not, so we still have 5 more minutes. We can ask more questions. There's a follow-up question for Youssef on your slides number 8. what is the similarity limit proposed for model independent MSD?
Yeah, sure. So that was muted. So actually, on slide 8, we applied the approach provided by Dr. Tsong in his publication in 1996. So assuming an acceptable difference of 10% at all the solution time points and with sample variance covariance matrix pulled across both batches, the reference and test.
Okay, great. So another question also to your slide number 7. So F2 is a biased estimate of F2. Bootstrapping F2 without correct the bias may lead to conservative result. Could you please comment on that?
Yes, sure. So actually, for this work, we conducted both the bias corrected and the BCA F2 bootstrap and the non-bias corrected. So regarding the conclusion on detecting or the sensitivity toward identifying the similar dissolution profile with theoretical F2 less than 50, actually both the BCA and non-bias corrected F2 bootstrap, the results were very similar. However, when tried to identify the similar dissolution profiles with the theoretical F2 between 51 and 52, we noticed that there is like a significant reduction in the passing rate when we use the BCA F2 bootstrap. So which means here, like using the bias corrected, could like the F2 means that here that without using the bias correction, there's more conservative behavior for the F2 bootstrap. I hope this answered the question.
Great, thank you. Yi and Meiyu, do you want to comment on that as well?
Yeah, I think the many of the publication of the bootstrapping method is correcting the bootstrapping itself because the distribution may not be symmetric, it's skewed, so they use the correction for that kind of bias. But the estimation itself is biased, as we mentioned in 1996 paper. So you need to correct the bias before you do bootstrapping. Otherwise, it's going to be conservative because we show that the estimate is the expected value of the estimate is smaller than the population value of the F2. So that's what we last year we wrote the paper, we wrote the manuscript submitted for publication to include a bias correction before we do the bootstrapping, and which shows the result is going to be improved for that.
Meiyu, do you want to add your insight as well?
I think I agree. We did a research for bootstrap method, we need to correct the estimation bias first and then do bootstrap.
Okay, all right, thank you all. I think we exhausted our time, so I will hand it over to the organizer. Thank you very much for your participation. Thank you.